Hi, folks. This is Dr. Pickett. I just wanted to do the second half of the Lecture 13 slide narrative for you folks. And I'll go ahead and post that as soon as it's done. I hope everyone is doing well today. So where we'll be beginning today is in Lecture 13, slide 29. Just as a little bit of a preface before we get started on this. So this chapter focuses primarily on post-transcriptional regulation of gene expression, i.e. regulation through phenomena like alternative splicing, RNA stability. Just as a cautionary tale, the authors here, or just to raise a cautionary point, the authors in your text are very enthusiastic about the somewhat controversial concept that the cellular complexity and tissue complexity of particularly interesting organisms like humans maps in some way to the protein complexity, the number of protein and type and diversity of protein molecules that are present in cells. I just point this out that this isn't a well-supported concept at this point. If we think about our friends in the chemistry department, our biochemists who study proteomics, who are able to enumerate, count, and describe the number of proteins that are accumulating in cells, there is in fact relatively limited evidence that, for instance, human tissues are more complex than Arabidopsis thaliana, a famous plant tissues, or are more complex than name your other organism, even some single-celled organisms like different algal variants. So I'm not exactly sure why your textbook authors are so enthusiastic in their embrace of this concept, but I just want to highlight for you, and I will also put some links to some, shall we say, countervailing interpretations. In particular, this chapter focuses quite a bit on the concept of alternative splicing, where with many exons and introns, a pre-mRNA can be alternatively processed based on different components in the spliceosome, the splicing machinery in different tissue types, such that in one tissue, perhaps all of the exons in the mRNA are used. In another tissue, there may be RNA interacting proteins or spliceosome interacting proteins or RNAs that interfere with splice donor and acceptance interactions. And so there may be situations where, for instance, very large introns are spliced out that contain within them exons. So alternative splicing is well characterized. I certainly know, even in the 80s, we had some good examples when I was an undergraduate, some good examples of alternative splicing as a biochemical phenomena. But recently with the onset of RNA sequencing, quote unquote RNA sequencing, the ability to very rapidly enumerate and describe many distinct RNAs isolated either from single cells or from ground up tissue. It's clear that our biochemistry is very sensitive and can detect RNAs that are present in a tissue, even a fairly simple tissue like liver or brain or tissue that's fairly cellularly homogeneous. Don't tell a neurophysicist or neuroscientist I said that. But that we can detect RNAs that are present in a tissue at a concentration of quite literally fewer than one molecule present per cell, i.e. in 10 cells, one of those cells may accumulate a specific mRNA, or in 100 cells, one of those cells. Well, we can detect that. So we're able here to detect a huge amount of RNA, which may in effect be biological noise. It may not be integrated into the physiology and biochemistry of the organism. Evolution may have no way of selecting for the benefit of one of these alternative mRNAs. So I just say that because I feel like I need to provide a bit of a counter discussion to balance the very strong statements that are made in your text in this idea that human cells have, hundreds and hundreds of thousands of distinct proteins, and that there may be individual enzymes that have 20 quite distinct protein forms present in a cell. And I just want to point out that the evidence for this is quite poor. It's a fascinating hypothesis, but as of this point, we do not know if what we can detect really has biological meaning. So I just wanted to point that out at the start because I feel like your textbook does come down very strongly on this model of biology that cells are chock full of proteins that are quite distinct from each other. And one, as a bit of an evolutionary biologist, I have a little bit of a hard time understanding how, for instance, in the short course of tens of millions of years, individual organisms could develop incredibly, some might say, wholesalely distinct proteomes in relation to each other. It just seems quite implausible on its face. And so I just want to point that out, that there's quite a bit of hypothesis testing and that needs to be done, and there's quite a bit of speculation in the text around this concept. I'm not a proteomicist, I'm not a biochemist, I'm a bit of an evolutionary biologist, so that's a bit of my perspective. But the sort of pre-test probability, if we want to go to that point, that human cells have hundreds of thousands of distinct proteins in them doing novel stuff, when somebody with a mass spectrometer cannot detect the presence of any of those proteins, seems a little bit problematic. And I don't want you guys to move off into your professional life misinformed about the biochemical complexity of the cells and tissues you'll be working with as physicians, et cetera. It is likely that if you identify a perfect inhibitor of one form of an enzyme, and that's the mRNA that's present at abundance of 99%, chances are very good you've identified an inhibitor that's probably going to be, if it could be developed into an interesting drug. So anyway, I hope that helps. Okay, so what I'd like to do today is start on, I'm sorry, pardon me. I would like to start with slide 29 in lecture 13. And now I just want to remind you again that this is a slide narrative. And so I hope you will follow along with me in the actual PowerPoint slides, which I've made available online. So what are we dealing with in chapter 18? Well, I've obviously let the cat out of the bag here a bit in my previous discussion. We're talking about post-transcriptional gene regulation. What does that mean? That means, at least when we're talking about alternative splicing, we are assuming that whatever's happened in terms of promoter activation, enhancers, activators, silencers, repressors is done, and a transcript is now accumulating. But then the next question is moving that pre-mRNA into a fully spliced form of mRNA, which is then available for translation into a specific protein. It is clear that regulation can reach into the flow of biological information, the if you will, retrieval of biological information, and can influence processes after a pre-mRNA accumulates to affect whether or not a functional protein is read out by translation. Now, if you have missplicing, this will often cause the activation of a cellular response called nonsense-mediated mRNA decay. If you have a splice form that cannot be translated appropriately and you have an early termination, that mRNA will tend to be unstable. So this can be an effective way of changing the level of transcript that's present in the cell, changing the level of protein that's in the cell by making sure that an mRNA is through an alternative splice or through a regulated splicing event shunted into degradation. So even if transcription occurs and pre-mRNA is accumulating, that does not necessarily mean that we can predict that a protein will be read out by translation, that post-transcriptionally splicing and other phenomena will produce what we would expect from the raw information that is stored in the genome. So the first example that they talk about is a very interesting type of alternative splicing that ends up producing two different small peptide hormones, if you will. They interact with specific receptors that are present in thyroid and also present in neurons. So the gene that they discuss, which is described on slide 33, figure 18.3, shows a coded form of information in DNA where we have 6 potential exons that are present, although a deeper dive is interesting. Because if you actually look at RNAs accumulated from this gene, you do see 2 fairly major alternative splice forms, depending on which tissue you look in. If you grind up some neuronal tissue, you see 1 splice form. If you grind up tissue from the thyroid, you see another splice form. And so this is good evidence that there is tissue-specific regulated alternative splicing having an impact on the regulation of this gene. That means there are different components of the spliceosome, different RNA binding proteins, et cetera, that are active in these 2 tissues that take the same pre-mRNA, but then process it differently depending on the tissue that it is in. So to begin with, we see encoded in the form of DNA, 6 exons with the appropriate introns. But what we see is that there is an early polyadenylation site and transcription termination site that is correlated with splicing exon 1, 2, 3, and 4 together. And we see the pre-mRNA spliced into the RNA in that form when we look at thyroid cells. So looking at an RNA generated from this pre-mRNA in thyroid cells, we see exon 1, 2, and 3 spliced together, the appropriate introns removed, and then finally exon 3 spliced in to exon 4, so that you have an entire reading region. The polyadenylation site is then used and you end up with a shorter transcript. When that transcript is translated by the ribosome, it ends up producing a longer polypeptide, which is then further processed into a peptide that's referred to as calcitonin, and that plays an important role in calcium ion concentration regulation in blood plasma. So this is a very important signal that helps the plasma maintain its appropriate concentration of that ion. However, if we look at neurons, what do we see? We see the same pre-mRNA accumulates, the same pre-mRNA is present. However, as splicing commences, we see that exon 1, 2, and 3 are spliced together, but the donor and acceptors completely ignore exon 4, and exon 4 ends up being released as a large lariat-formed intron that we presume is eventually degraded and destroyed, and exon 3 is spliced into exon 5, which is then spliced into exon 6. So in this particular splice isoform or splice variant, we see 1, 2, 3, 5, and 6 in terms of the exons that are spliced together. After processing, polyadenylation, and capping, that mRNA is further translated into and then post-translationally processed into another peptide called the CGRP peptide, which is quite fascinating because although it is produced by neurons, it plays a role in smooth muscle and other regulation involved in vascular blood pressure. So it ends up causing a change in the vessel diameter that's associated in particular in the brain and other regions. So this is quite a fascinating example where tissue-specific regulation of alternative splicing plays a very important role in generating two different proteins from the same pre-mRNA, but not from the same codons. In one case, only the codons of exon 1, 2, 3, and 4 are used. In the other case, codons from exon 1, 2, 3, 5, and 6 are used. Both of these alternatively spliced mRNAs have distinct polyadenylation sites. They produce transcripts of different lengths and transcripts with different sequences. In common, towards the 5' end of the transcript are the first 3 exons, and those codons are shared. However, after the first 3 exons, both the codon sequence and the amino acid sequence diverges as you get more towards the carboxy termini of these two different proteins. So this is an extremely well-characterized situation where alternative splice forms, alternative types of mRNAs, produce distinct types of proteins which have different roles, calcium homeostasis or changes in terms of vascular dilation in different tissues based on their unique roles in different parts of the body, their unique protein accumulations in different tissues. Now, so the existence of examples like calcitonin and the CGRP peptide certainly led to speculation that alternative splicing might be broadly seen in the genome. Older editions of genome-wide databases showed many, many human genes with a large number of alternatively spliced transcripts. More recent iterations of those genes with sort of further curation for different types of functional criteria. A lot of those transcripts have vanished from the databases. So, you know, how widely distributed this really is as a phenomena is, as I've said, still an open question. Another situation which I think is quite fascinating where we do see alternative splicing, if we move to slide 34 in lecture 13, is the phenomena of the Drosophila cell adhesion molecule DSCAM. So this is quite a fascinating gene and certainly complex pre-mRNA. Kind of neuron to neuron, axon to axon, you see in Drosophila different splice forms and different splice variants accumulating, but also distinct tissues appearing, also distinct proteins appearing on the membrane surface of these axons. So how much complexity do we get with this individual gene? It's quite fascinating. If we look at the exons that are variant, that are distinct from cell to cell to cell, and if we also look at the little library of exons that are present stored in the DNA for this gene, we see quite a fascinating story. For instance, if we look at exon 4, we see that there are 12 alternatives of exon 4 encoded in the DNA, but any individual mRNA will only have one of them present. For exon 6, we see 48 alternatives encoded in the DNA. Exon 9, 33 alternatives. Exon 17, 2 alternatives. So what's quite fascinating about this is in a pre-mRNA, we see all 12 of the exon 4 alternatives that are present, all of the exon 6 alternatives, all 48 are there. So these transcripts are gigantic, but then in the processed mRNA, we see only one of the exon 4 exons are present. Only one of the 48 possible exon 6 exons are present. So here, alternative splicing has been playing a really, really important role in choosing one set of codons from amongst many at different sites in this gene. So you might ask yourself this, I think, very fascinating question, how many alternative forms? They think at this point, you may be able to generate more than 30,000 distinct forms of this type of protein based on these alternative exons, alternatively spliced exons. So this is a fascinating phenomenon. Now ask yourself this question: why? Why do we need such a huge amount of protein diversity? Well, it turns out these DSCAM proteins are quite fascinating because they interact with each other and bind. So based on the exon 4, 6, 9, and 17 forms that you have a tendency to bind to the same isoform type. However, if there is a difference, those DSCAMs do not bind as well to each other. So this suggests that there's a lot of regulated splicing so that an axon kind of growth cone working through tissue, when it encounters another tissue, it's going to use the DSCAMs on its surface and their similarity to the DSCAMs on the surface of the target to make a good axon contact with whether it's an interneuron or other type of contact. So this is quite fascinating. This high level of diversity is probably required for wiring up the Drosophila nervous system in a fashion that is functionally appropriate for the organism. So these are two very nice examples of alternative splicing where we are clearly getting a bit of protein diversity. In the case of our little calcitonin peptide situation, we have two alternative splice isoforms, two protein isoforms. In the case of DSCAM, we may literally have tens of thousands of distinct protein isoforms that form. As I've said previously, this does kind of lead us to ask the question, how universal is alternative splicing? And I will say, as I've said previously, this is a challenge for your generation. I hope you guys find out over the course of your long scientific careers. My guess is most genes are not producing 32,000 isoforms. Many genes are probably only producing one isoform, but this is an informed guess at this point at some level. I won't oversell it, but I also think it's quite a fascinating venue for future study. Okay, now, one thing that I think that's interesting about alternative splicing in the situation where we have very well characterized examples is kind of what sort of phenomena is it integrated with. One of the best characterized genetic networks that it is involved in is Drosophila sex determination, where alternative splice forms and splice forms in a genetic pathway where you have splicing leading to splicing, leading to splicing, leading to function, or alternative splice leading to other function. This is strongly integrated into Drosophila sex determination. So one pattern of splicing results in male sex, the other pattern of splicing results in female sex, and regulated alternative splicing with the production of sex-specific isoforms of mRNAs and proteins is absolutely required for Drosophila sex determination. So I'll direct your attention to slide 36 at this point, where we talk about the female and male sex determination pathways and the alternative splicing that regulates sex determination. Now, sex determination is something we haven't discussed in depth, particularly for Drosophila. We talked a little bit about testis determining factors, SRY, et cetera, in mammals, particularly in humans. But in Drosophila, it's quite a fascinating situation. The two sexes in Drosophila are somewhat similar to what we see in humans in terms of their chromosomal sex. Females are XX and males are XY. However, females have a system that's a little bit different in term, in female mammals, for instance, you have X inactivation, and so there is a phenomena where for many genes on the X, you have lower levels of expression. And so that's how we have sort of a balancing of the influences of the X chromosome, male to female in humans. Males do not have any silencing of the X chromosome. All of their X-linked genes are expressed at the levels and in the tissues that they should be, whereas females, through random X inactivation, have a tendency where on a cell-to-cell basis, they will have one active X, one less active X due to silencing. So Drosophila cells are able to essentially measure the quantity of X chromosomes that are present within them. And if two X's are present, you start a female sex determination pathway. If one X is present in terms of the X to autosome ratio, you start a male process of development. In females, there is an RNA binding protein called sex lethal, and its transcription is activated by the X to A ratio, where you have 1:1, two X's to two autosomes per autosomal chromosome. Sex lethal protein accumulates. It is an RNA binding protein, and it interacts with the target RNA transcribed from a gene called transformer. This causes a female-specific splicing pattern to occur that causes transformer protein to accumulate in females. Transformer protein is another RNA binding protein that in turn targets a double-sex mRNA. This causes a female-specific splicing pattern to occur in the double-sex mRNA, which produces the female form of double-sex protein called DSXF, using the nomenclature of the authors of your book. DSXF protein also then causes female differentiation. So if we contrast that to what goes on in the male cell, where the X to autosomal ratio is not 1:1, but is 0.5, because there's only one X per any two autosomes that we're looking at. If you have a 0.5 X to autosome ratio, you do not activate sex lethal transcription. Thus, no sex lethal protein is translated. Since sex-lethal protein is not available, although transformer mRNA does accumulate, the female-specific splicing pattern does not happen and no functional transformer protein accumulates. So if you have sex-lethal, transformer undergoes alternative splicing and transformer protein accumulates. In the absence of sex-lethal protein, the male-specific splicing of transformer occurs and no transformer protein accumulates. In the absence of transformer protein, alternatively, again, the double-sex pre-mRNA is now spliced into forming a male-specific form of the double-sex protein, and that double-sex protein then causes male transformation. So this is quite fascinating. In females, you have a unique female-specific RNA binding protein that accumulates. It causes a female alternative splicing in terms of its target. That produces a female-specific protein, which then causes an alternative splice of the ultimate target. And so we have an entire genetic network where there is a female-specific regulatory cascade, all mediated by alternative splicing, except for the initial transcription of sex-lethal protein, in contradistinction to a male cell where sex-lethal protein does not occur, and so you have a cascade that leads ultimately, in effect, through the absence of alternative splice factors with the splicing of the double-sex protein into its male form. Cells that accumulate female double-sex feminize, cells that accumulate male double-sex masculinize, and so it's quite a fascinating process where in this organism, in this specific biological process of sex determination, alternative splicing has evolved to play a major role in that kind of genetic switch between female and male determination. So it's quite fascinating in this case because sex-lethal is essentially acting as a genetic switch, which causes a cascade of alternative splicing events that leads to female sex. In the absence of sex-lethal, that cascade of alternative splicing does not occur, transformer does not accumulate, and the male form of double sex does accumulate. Okay, so let's transition to slide 38. Moving away from alternative splicing as a phenomena or process, now let's talk a little bit about regulated RNA stability. I just want to very briefly, as your book does, kind of introduce two alternative processes that can be used to kind of modify the steady state level of mRNA. Clearly, we can have regulated instability of the RNA. You can have RNA degradation, which is targeted. So if RNAs are broken up as the concentration of functional RNA drops, the concentration of translated protein must be dropping as well. This can be a very effective way of bringing ultimately a protein below its concentration threshold for whatever biological process it is required for. We'll talk a little bit about how RNA-RNA complementation and hybridization can mediate that in just a bit. There's also evidence of some feedback between detecting concentrations of RNA and activating or deactivating transcription as well. So it's interesting. There can be RNA stability, which is a level at which we regulate whether or not a protein is above or below its threshold requirement for function, or we can have feedback kind of assessing concentration of RNA and whether or not transcription is occurring at the same rate it was previously. So I direct your attention to section 18.3 on slide 40, RNA silencing. can control gene expression in several ways. And it's quite fascinating because this is mediated by RNA-RNA complementation, where you have a very short stretch of regulatory RNAs. These may be called interfering RNAs, microRNAs, small interfering RNAs. Often they are produced from short transcripts that have self-complementarity, which are then further processed into these short targeting RNAs. Once they are present as a short targeting RNA, they can be complementary to a target mRNA and can then have their functional role. So this is kind of sequence-specific, post-transcriptional, but RNA-RNA complementation dependent. And this phenomena has been referred to as RNA-induced gene silencing. RNA interference, if it is effective in terms of regulating a gene or is used in regulating a gene, it does require a protein partner to interact with the short stretch of RNA. That protein partner is called dicer. It will cleave double-stranded RNA molecules into these small interfering and microRNAs. They can both repress mRNA translation simply by having pairing between the target mRNA and the small interfering RNA. That can, we believe, potentially by physical exclusion, prevent the ribosome from translating the mRNA that it is part of, or maybe even associating with it, depending on where the small interfering RNA is interacting with. The two processes that your book discusses is RITS, RNA-induced interference of transcription, and also RNA-induced silencing through RISC complex. Figure 18.7 kind of describes these two specific situations. The question we have at this point, though, is where does this kind of double-stranded piece of RNA that's going to kind of be the targeting molecule in this process come from? That little piece of double-stranded RNA might be delivered from something like a virus, a transposon, or even a synthetic RNA that we make in the lab, or it may be transcribed from a microRNA or small gene, where a small self-complementary piece of RNA forms that then can be further processed. Whatever it is, you need a short stretch at least of self-complementary RNA that is then fed into the dicer protein. That will cut off any extraneous pieces of RNA and process them into these very short, either siRNA, self-complementary short helices, or miRNA, self-complementary helices. If those short pieces of RNA are then fed into the RISC complex, the RISC complex will then kind of move to the target mRNA and degrade it. That drops the concentration of functional RNA, and with less RNA, you have less protein. Or if these small pieces end up being associated with the RITS complex, that can end up causing the RITS small RNA complex to cause chromatin-mediated silencing or other types of influences on transcriptional initiation. So these two processes are quite fascinating because here, again, it's a post-transcriptional regulation. RNA is accumulating. It's either being targeted by a small RNA for degradation, or it's being targeted as part of the RNA polymerase complex to eventually cause inactivation of a gene through chromatin remodeling. But in both cases, transcription is occurring of the target mRNA, and these regulatory RNAs are also acting. So I know this is a fairly short, rather quick summary of post-transcriptional, if you will, gene regulation. the main thing I wanted to do with a discussion of this section is just highlight that as fascinating as alternative splicing is, its real role in the cell is somewhat controversial. Many claims being made for it in terms of complexification of the biochemistry of organisms are yet unproven. And in fact, although we say, absence of evidence is not, no, evidence of an absence is not, I'm going to mess that up. Evidence of absence is not absence of evidence. No, absence of evidence is not evidence of an absence. That is simply because we don't see evolutionary selection of alternative splice forms, or we cannot detect the presence of alternative splice proteins accumulating. That doesn't mean that alternative splicing isn't important. It's just the cases where we really know what its role is, very fascinating. Speculation about how broad its role is just that. It is scientific speculation. As we get more and more evidence, we may come to embrace the idea that the proteome is extremely complex in different organisms, or we may have to reject that hypothesis as things move along. Thank you very much for your attention, my friends. Have a good day.